we are not solely responsible for how we think, feel, and act. Bacteria in the human body comprise roughly 10 times the number of our own human cells, and 80% of the bacteria are in our gut. These bacteria can have influences on our bodies and minds. Our microbiome population, which includes bacteria, viruses, protozoa, archaea, and fungi, are constantly fluctuating, depending on what we eat and what's in our environment. These microbiomes are everywhere in and on the human body. The gut microbiome has various methods that affect us, and what you may not know is that we communicate with these gut bacteria, and these gut bacteria also communicate back with us through complex pathways in our nervous, endocrine, immune, and metabolic systems. At birth, the initial microbiome of the baby comes from the mother. Whether it's a C-section or a normal delivery, the microbiome is impacted by the surrounding skin bacteria from which the baby was delivered. After a year, the microbiome matures to about what an adult has. Gut bacteria have bidirectional communication with the human body through the gut-brain axis. Gut-brain axis uses the nervous system, endocrine system, immune system, and metabolic systems to change dynamic bacteria populations and human body molecules. Through the nervous system, the lining of the intestines has cells communicating with nerves around the intestines. These nerves then communicate to the autonomic nervous system of the spinal cord, which communicates to the brain. Thus, interference by bacteria in the communication between any of these areas could cause intestinal function changes. Through the endocrine system, bacteria can produce molecules that can have either beneficial or negative effects on our bodies. These potentially beneficial molecules include dopamine, which is an important neurotransmitter, gamma amino acid, an essential molecule for preventing diseases such as neural diseases, butyric acid, a suppressor of colon cancer, short-chain fatty acids that regulate metabolic processes, and serotonin, essential for memory processes. And 95% of all serotonin in the body is produced by gut bacteria. But some bacteria can also make neurotoxic D-lactic acid or ammonia, which the human body has to either recycle or eliminate as it is unhealthy for us. Hence, the human body can be linked to the composition of bacteria in the gut, and all bacteria in the body have a role to play. Bacteria microbiomes function to prevent diseases through various methods. Good bacteria function as intestinal barriers from dangerous microbes by colonizing and dominating most areas in the intestines, inhibiting the bad bacteria's foothold in the gut. This is called protective competition, and it can help reduce medical conditions like inflammatory bowel diseases, allergies, and obesity. For human structural function, bacteria benefit us by promoting the regeneration of human gut cells. They produce mucus for the gut, and produce short-chain fatty acids that nourish the human gut cells. In addition, gut bacteria help bodies metabolically by producing vitamin K, supplying essential nutrients, eliminating toxic molecules, and helping us digest cellulose, forming new blood vessels around the gut, and helping out our enteric nervous system, which is our intestinal nerves. Bacteria in the gut benefit our immune systems, especially early in life. Our immune system is challenged by the bacteria that enter our gut, thus preparing the immune system for future invaders and keeping a steady supply of immune fighters in the body. Bacteria and animals interact through emotions. When applied to rats, there was a study where a rat separated at birth from their mother had reduced lactobacillus in their microbiome later in life. This shows that stresses linked with separation reduce some types of bacteria in the gut. Other studies have shown that when mice lacking all bacteria in the gut are given stress stimulus, the stress hormones corticosterone and adrenocorticotropic hormone increase. These stress molecules could be reversed at varying degrees with bacteria microbiome transplants, especially with Bifidobacterium impetus. This demonstrates that stress can alter the bacteria in the gut, and the gut bacteria can also alter the stress in the body. When applied to humans, if a baby experiences stress in the first few years of birth, its microbiome could be altered. It was found that the risk of the baby having stress-related disorders later in life increases as a result. The dominant bacteria in our gut are the Firmicutes and Bacteroidetes, representing around 90% of the bacteria in the healthy human gut. If the Firmicutes-Bacteroidetes ratio is increased, then this could increase the likelihood of obesity in humans. Disturbances to the gut balance are called dysbiosis events, and this has been linked to these health concerns and more. In May 2000, Walkerton, Canada had a contamination of E. coli and Campylobacter jejuni in drinking water. There were around 4,500 people affected. Half of these individuals were assessed 8 years later, and half of those people assessed were linked to irritable bowel syndrome. A field that is working to unravel the mysteries of the microbiome is called metagenomics. Metagenomics takes the genome of microbiome populations, sequences these genomes, extracts their DNA, makes DNA libraries, analyzes the genetic composition in microbiome populations, and from all this, progresses to understand the relationship between humans, microbiomes, and the environment they're in. This field is especially important for advancements in medicine, for treating human diseases like cardiovascular disease and obesity. Ultimately, how we think, feel, and act are influenced by bacteria whether we like it or not. Whether it be through disturbances in the nervous system, hormones produced, or whether we seek medical attention for increased risks, it is important that we consider what we eat and what our environment has, as dysbiosis of the microbiome could have profound impacts later in our life.